Welcome to the Metabolic Link, a podcast that explores the common thread of metabolism in health and disease. This is where science meets society. Welcome to the Metabolic Link. I'm your host, Dom D'Agostino, a professor in neuroscience and physiology, and today we are diving deep into the world of mitochondrial health with an incredible guest, Dr. Anna Andreazza. She is a professor at the University of Toronto and an expert in mitochondrial function. Dr. Andreazza has pioneered research linking mitochondrial dysfunction to psychiatric disorders, as well as innovations in organ transplant, regeneration, and novel therapeutics for mitochondrial health. A key area of her study is the investigation of mitochondrial function in mood disorders like bipolar disorder, where she has identified a dysregulation in mitochondrial genes, and she's used 3D brain organoids to investigate how mitochondrial dysregulation impacts neurotransmission in the disorder. Ultimately, these studies are really paving the way to personalized treatments in mood disorders. Dr. Andreazza is also a pioneer in mitochondrial transplantation, exploring the stability, the safety, and the efficacy of mitochondrial transplants for things like regenerative medicine and chronic diseases. In this episode, we're going to explore how the mitochondria impact the mood, metabolism, and overall health, and why optimizing mitochondrial function is the key, or might be the key, to more groundbreaking treatments in the future. Before we dive into this podcast, though, I want to say a special thanks for our sponsors supporting this episode. We really believe in all the brands that we support. We use their products. I'm using their product now, and they make this kind of content possible for you guys to listen to. So thank you for engaging with them, and by doing so, you're also supporting us at the Metabolic Link, so we can continue bringing you cutting-edge research or science outreach on various research topics, metabolic health and therapy. If you prefer to listen to this episode without the ads, please check out our platform, the Metabolic Initiative, and we offer a comprehensive library of metabolic health therapy content, as well as we have private ad-free feed episodes. So visit membership.metabolicinitiative.com to learn more. Now, please enjoy my interview with Dr. Andreazza. Welcome, Dr. Anna Andreazza. It is so exciting to have you here on the Metabolic Link podcast. And I was really inspired by your work in Santa Barbara a few years ago, where you were spearheading the entire field, at least in behavioral health and in many other areas I want to talk about in mitochondrial medicine or metabolic medicine. And I would love to maybe hear your backstory and how your career trajectory got you into this field. And then I have a lot of other questions beside that.
It's such a pleasure to be here, and thank you for having me on this podcast, which has had so many wonderful interviews. So I feel very honored to be part of this team of interviews. My story started actually a little bit not in psychiatry at all. Actually, I was studying undergrad in biology. And my family is originally from Italy, and they immigrated to south of Brazil, where I grew up. And my grandfather, for father's side, had opened a winery a long, long time ago. And he was extremely, always frustrated, him and his family, that the wine wouldn't taste good. And one day, and take this with a grain of salt, because I was 60 years old, and I read this news on the local newspaper saying that they have found this compound called resveratrol in a wine. And this professor called Miriam Salvador in our Institute of Biotechnology, my little hometown called Caxias do Sul, was studying this compound. So me, with my naive, scientific, but eager mind, went and knocked on her door without no experience whatsoever in research, and said, my grandpa is really, really frustrated with his wine quality, but it's really bitter and it's not a really good wine. So I'm wondering, can we test these seeds? Perhaps it's full of resveratrol. And she opened the door of actually her lab and really brought me into science and research. And through her, I learned and discovered what is antioxidants, what is mitochondria, what is metabolism, and they start to become fascinating of how a lot of the functions in our cells were dependent so much on energy metabolism, and how much you could change a cellular fate if you actually change a cellular mechanism of energy production. And studying with her, I realized that was at that time, this is quite a while ago, there was not a lot in studying brain and metabolism of oxidative stress and antioxidants in the brain and even mitochondria in the brain. And I start attending more lectures during my pharmacy degree on psychopharmacology and trying to understand more this connection potential there. So I met a very interesting and very bright psychiatrist called Flávio Kapusinski, who is in south of Brazil as well. And I asked him in one of his lectures, are you aware of anyone studying this part of oxidative stress and mitochondria in brain, and especially in psychiatry? And he's like, what is this? So we become actually friends, and he invited me to do a PhD under his supervision, and we published one of the very first papers looking into oxidative stress markers in patients with bipolar disorder. So that's how my career really started, understanding first what is antioxidant, what is mechanisms, what is mitochondria in cellular fate, and then moving into bringing that knowledge and applying what I become extremely fascinated for me, which is the story of mitochondria and energy metabolism in patients with bipolar disorder.
Mm-hmm. And just so I know, what date was that? Not to date you, but, but I just want to, I'm kind of putting it in the framework of what I remember.
Yeah.
I have all your publications, but I forget what year that was.
So the first time that I started working with antioxidants was 2003. And the first publication that came out with bipolar disorder and oxidative stress was 2008.
Okay, so you've been working in this area, yeah, for quite some time.
Yes.
So the antioxidant research, and did you also do research on lithium too?
I
Was
did.
that, yeah, because I remember seeing a paper on that. I just made a note to circle it. Yeah, ask her about lithium. Yeah. So,
Yeah, and that was actually a lot of part of my PhD. We were at that time working with a model of what we call a mania-like model, which was inducing mania by increasing dosings of amphetamine on this, which is a really not an ideal model of mania, but it certainly was at that time, back in the early 2000s, were one of the most famous models for model of like mania. And we started looking into lithium. Lithium, of course, was reversing that effect, but then we started looking whether lithium could potentially be reversing that effect of amphetamine, because it was restoring mitochondrial function and decreasing the oxidative stress in the brain. And that's a part of what we founded. And I think that started leading at people to think, okay, if lithium is an effective medication for bipolar disorder, and it does has an effect on energy metabolism, perhaps these two things are linked to the pathophysiology of the disease. And then that starts spiraling on many others that came to the field to start bringing stronger evidences showing the link between mitochondria and the bipolar disorder.
And in the years after, you've really developed, I remember you talking about a number of innovative models, including the organoid models. And but before we get to that, because I was just measuring my lactate, I have a lactate meter here. It's something that we do actually in our cancer models. And you had made reference to there's off-the-shelf technologies that allow us to do, the single biomarker. And I
Hmm?
remember in your talk that you highlighted, and you have a publication too, highlighting lactate as a clinically relevant biomarker for metabolic health. And maybe discuss a little bit about the research you've done on that.
Yeah, that's a fantastic question because I
Mhm.
think that brings one of the, in my perspective, one of the most convincing points why metabolism is so important in medicine.
Mhm.
So if you look back in the 1954, Herman have published a paper showing high levels of lactate in that time comanic patients. And this is a field that has grown over the years, and we keep like doubting the data, right? We keep saying, oh,
Mhm.
maybe we need more samples. Oh, maybe we need to redo this. But this is not connected with the young men rating scale. It's not connected with the cognitive findings.
Mhm.
And continue with finding exactly the same thing. Over 10 publications using magnetic resonance spectroscopy in the brain showing increased levels of lactate in the brain. Several publications showing, including our own publication, showing high levels of blood levels of lactate in patients with bipolar disorder. A very recent publication just of the beginning of this year, we partnered with Marion Leboyer at Fondation Fondamentale in Paris, France, and looking over 800 patients with bipolar disorder, the levels of lactate, and compare this with the presence or absence of metabolic syndrome. And that patients that had well-characterized metabolic syndrome had higher levels of lactate compared to those that don't have. So a very strong, interesting point to say this is not relevant to all the patients, but really strong to those that actually have the metabolic syndrome association with the disease.
Mhm.
So we went a step further and now did a mitochondrial metabolomics in these patients and identify that if we stratify these patients by those that have levels of lactate higher than 2 and levels, and they have a completely different profile of a metabolism than those that have a normal level of lactate and no metabolic syndrome, and also those that have normal level of lactate but metabolic syndrome. So
Mhm.
understanding that, it starts telling you a little bit about how to treat these patients, because you can find,
Mhm.
well, these patients have much more alpha ketoglutarate, which
Mhm.
you're probably going to start thinking, well, so maybe a ketogenic diet here will be quite an interesting in these patients. They have high levels of citric acids, so involved
Mhm.
with cholesterol, so maybe potentially statins and metformin, so be other treatments. So how to treat these patients can be highly informative. And you said this in the beginning, and I emphasize this. These are available today markers that cost almost nothing to be done in our
Mhm.
patients. Cholesterol,
Mhm.
insulin, belly measurement, waist
Yeah.
circumference, lactate, it's all there. So it's not something that we need to wait for FDA
Mhm.
approval, validation. We can start taking action and convince our medical educators and medical professionals to bring this to more light on the front of the patients and just take a look. Potential is not there for your patient, but if it's there, it could really help you guide it on your treatment.
Genova Connect is a sponsor of this episode. Want to learn more about your own health? Genova Connect is powered by Genova Diagnostics, and they offer a very simple at home advanced lab testing kit that's backed by science and really takes the guesswork out of understanding your own health. I've been a huge fan of Genova products, including the Metabolomics Plus kit. So this is it uses a urine and a blood spot samples, and it assesses things like oxidative stress. It assesses mitochondrial function, omega-3 levels are also assessed in the kit, and the ratio, for example, of omega-3 to omega-6, methylation balance. There's a broad list of toxins that is also measured by the tests. And I often put their data that I acquire personally into my own presentations, so academic presentations or invited presentations. So I really love their tests. It's very informative, and many of the things that are measured give me great insight into my own metabolism. Genova Connect is offering 50% off any of their test kits when you use the code Metabolic Link at gdx.net/the metabolic link. Now back to our interview with Dr. Andreazza. A question with the bipolar, did the lactate correlate with the manic phase? Was there a particular phase? Like there's the hyperglycolytic sort of,
Yeah.
hyper excitability sort of
Yeah.
hypothesis out there. Yeah.
Very, 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 very good question. We did not find
Mhm.
a very strong correlation with the young man rating scale. Yes, it does has a better bit of a correlation, but you have to keep in mind that this 800 over 800 patients were outpatients. And for
Mhm.
these outpatients, they don't really have a very big range on your scale. And so they are mostly well treated, which we call in euthymia. Right? They are
Yeah.
very well. We then evaluated on a secondary cohort, which is called the I-give cohort, also from Paris, France. And that's a much smaller cohort. But yes, in those patients that they were inpatients, that they were in the hospitals when the blood was collected, that if they were over mania, then yes, the lactate levels were much higher, and
Mhm.
that is an association potentially with
Mhm.
phase and state of the disease. But we needed to replicate this data on a larger cohort, in my perspective, to really have. I think the bottom line for me, the learning over the years, is independent of whether this is associated with a phase of the disease, it is certainly associated with the progression of the disease and
Mhm.
how these patients will be treated over the disease. Because we know that a lot of medications that we give to patients with bipolar disorder will further enhance the metabolic dysfunction in these patients, will further damage the mitochondria, increase
Mhm.
lactate further, so the patient continue going the spiral down and not getting the treatment that will help them to not achieve that high level cardiovascular disease we see in our patients and many other diseases like diabetes that are often associated with bipolar disorder.
Yeah. It just occurred to me because I was doing some tests myself, where I took metformin, and metformin is used for type 2 diabetes. I had a PhD student who did his dissertation on it, and it's a weak mitochondrial toxin, and we measured superoxide production, is increased with metformin. But metformin can help to improve
Yeah.
so it's a weak, metabolic toxin, probably at the level of the liver, and contributes to acidosis. And
Yes.
I'm wondering your thoughts. I didn't intend to ask about this, but
No, no.
on metformin, and
Yeah.
it does elevate lactate, and could that be contributing, or what's your thoughts there?
It's a very interesting, and it's a really, really interesting question, because we have to be careful on when we talk about this, because we're always thinking about the measurements as a static measurement. And as you said, you took
Mhm.
the metformin and you measure the lactate, you see a
Mhm.
difference. But
Yeah.
over time, that actually will change if you continue measuring, right?
Mhm.
So that the way that the mitochondria respond to each other from one cell to another is by signaling stresses and sending molecules around. So
Yeah.
in our brain, for example, you have something really important, which is the lactate shuffle.
Mhm.
Right? You need to pass the lactate from the astrocytes to the neurons. So that temporary increase of the lactate, if it is temporary, is actually a good thing, because it's passing to the neurons and lactate is entering the neurons, and now we're going to actually act as an energy source. What becomes problematic is actually if this becomes the state of art of using the energy production in your brain. because you're not going to have the same amount of ATP production that you have if you have an efficient oxidative phosphorylation ongoing, right?
Yeah.
So that's one of the things that we always keep thinking, could this be a way that the ketosis is actually helping the brain to rebalance itself? Because you're increasing the presence of ketones. Ketones are also used as an energy coin in your cells. It helps to produce ATP in your cells. So could this be easing down this lactate necessity of production? And when you're asking about the metformin, is the metformin trying to also potential for being a mitochondrial weak, pushing into the lactate to kind of compensate that is more inhibition that the metformin is well known to do on complex 1? So could that be just a compensatory mechanism? And again, if it is a temporary compensation, potentially not a toxin that is very strong. But if you put the metformin now on someone that has a complex 1 dysfunction, that could be a problem, right? So I think everything depends on identifying the right treatment for the right person at the right time. And that's one of the things that I think is the next generation of a metabolic medicine that we're going to see is more precise profiling and understanding how to target the best treatment for you.
Yeah, and very, very good points. And yeah, context is very important here. And metformin is also very pleiotropic, so it does a lot of different things on insulin sensitivity and whatnot. But it just popped into my head because the same experiments we were using metformin in, we were using something called dichloroacetate, DCA, which activates PDH, and that's specifically used for reducing lactate production. And I don't know if anything, if you're aware of that drug, it's actually was used quite by a group in Canada for GBM and other things for a therapy. And we've tinkered with it in our lab a bit, but it just comes to mind because that can dramatically, lower lactate. And could lactate lowering therapies or mitochondrial enhancement, that also coupled with thiamine, I think B1 sort of stimulates the function of the pyruvate dehydrogenase complex. And I think some people have looked at that as being the bottleneck, as being sort of the rate limiter for glucose oxidation.
And so I think you said the word that is the most important. Context is everything. And actually finding that's why we have to be a little bit careful when we say, okay, it is really high lactate and let's treat the lactate. Sure. If it is at 5, you need to bring it down. That's well known, right? But if it is in the levels of 2 and 3, how do we treat that? I think we need better context to understand what is leading to that patient over time to actually lead to that accumulation of the lactate. So maybe it is a PDH inhibition, then you have maybe actually going into lower treatments of lactate is actually good. But if it is actually even above that on the glucose pentose cycle, so should we be treating differently? Should we be treating insulin resistance to actually treat the lactate accumulation on this patient? So I think that kind of level of granularity, I think, is what we're going to be seeing in the next few papers and the years of study, and really hopefully in a much fast progression that as we have progressed from here to really precisely help the patients. I just want to be careful that we don't, I'm trying to not say because you have this 2 and 3, go on and treat it and decrease your lactate and everything is solved. That's absolutely not the case. We need to understand the context of what is causing, what is coming, what is the pathway to properly address and properly treat the patients.
Yeah, and that's a good lead into sort of some of my next questions. Looking at, you had talked about in your presentation about mitochondrial DNA and mitochondrial mutation, and circulating mitochondrial DNA and its correlation with bipolar. So I would love to hear some of your thoughts,
Yeah,
on that field of it's a nascent area of research, I think, and you're
I
spearheading it.
think
And
so.
yeah.
Yeah. So circulating mitochondrial DNA in the field of psychiatry is a little bit of a starting point for us to
Mhm.
understand how is it role. But circulating mitochondrial DNA in cancer, for example,
Yeah.
it's a well-known marker.
Yep.
They use actually currently and routinely to actually understanding mitochondrial toxicity of some of the chemotherapies, right? So
Mhm.
what is a circulating mitochondrial DNA? Our mitochondria are very tight and well packed. They have double membranes, and the DNA that is inside the mitochondria should not escape, right? It should stay
Mhm.
there and is actually should be very well protected. When
Mhm.
the mitochondria suffer stresses or outside stressors, the mitochondria form porous, and the DNA can actually leak out of the mitochondria, leak out of your cells, and goes to the circulation. And
Is
that's
that
what
the, sorry to interrupt, is that the mitochondria permeability transition, the MPT pore, or mitophagy? So,
it
uh,
could be both.
mhm.
So there are papers supporting both. The pore transition will release some of under the stress conditions. It releases the circulating mitochondrial DNA, portions of that DNA out of the mitochondria. But autophagy also, when the mitochondria, the cells will go, it could release as well. So
Mhm.
difficult to differentiate where
Mhm.
the circulating mitochondrial DNA is coming from when you measure in live humans.
Essential fatty acids are crucial for brain function, hormone optimization, immune response regulation, skin health, and proper cardiovascular function. C15 is the first essential fatty acid discovered in 90 years. Low C15 levels can wreak havoc on your metabolic health, and higher levels are linked to many health benefits we've discussed actually in a previous podcast. And what's interesting is that without even realizing it, most of us are deficient in C15, or it's not optimal. And this can significantly decrease our energy levels and resilience in many different ways. Fatty 15 is a science backed, award winning, pure C15 supplement. And if you are not eating a lot of animal products, for example, if you're more plant based or vegan, you are definitely most likely deficient in this fatty acid. I personally found that my C15 essential fat level was in a good to normal range, but maybe not optimal. Not surprisingly, others have told me that they now supplement C15 due to testing and showing a clear deficiency in this essential fatty acid. So you can get an additional 15% off their 90-day supply subscription starter kit by going to fatty15.com/metabolic link and using the code metabolic link at checkout. Now back to our interview with Dr. Andreazza. I was going to ask, can you determine whether it comes from, I mean, because you're measuring peripheral mitochondria, but it's
Mhm.
you're linking it with bipolar. And if you were to get circulating increase in mitochondrial DNA from the muscle, can it distinguish that?
Yes,
Yeah.
so
Okay.
that studies like that have been done beautifully in the area of cancer, where they actually have biopsies and they actually go directly into if it is a tumor or tumor area, surrounding areas, muscles, they compare and it has beautiful results showing you can actually determine the source of where it is coming. I am unaware on psychiatry that we have actually done that on that precision to determine the source. What
Mhm.
we have taken a little bit of a step further was to at least understanding from transforming the white blood cells of the patients into an iPSC, induced pluripotent stem cells, and then using that stem cell to generate what we call a brain organoid, but that's honestly this is it's just a little bulb of cells that represents your brain cells. So it's very
Mhm.
important to remember that the organoids that represents our brain cells, they don't have a formation of a brain. They don't look like a brain. They can represent cells of an area of the brain. We can have midbrain organoids that represent your dopaminergic
Mhm.
neurons. You can have glutamatergic driven organoids, or you can have a mix of everything, which is called cerebral organoids.
Oh,
So
okay.
that's
So just to clarify, so
Mhm,
you are taking
mhm.
these iPSCs and creating these brain organoids
Yes.
that that have a phenotypic identity, the dopaminergic, serotonergic, GABAergic, and do the cells and the organoids have processes? Do they fire action potentials? Do they have, okay, so they have voltage gated sodium channels, and you can
Yes.
do electrophysiology and measure action potentials. Okay.
Yes.
Very
So
cool.
if to be able to say that you have an organoid, you actually do have to do stains for different types of um, neuronal types and the glial types. If you go into 9 months, you actually start seeing astrocytes. Um, you have to do electrophysiology to show that you actually have fine potential, gate potential. So yeah, so that you do. What is this model good for? This model brings the genetics of the patient, right? So you can now take that genetic that is actually helping to create those cells and start testing many different things to see what stresses the system and what works better in the system. So it's a platform, if you like to put that way, to understand better pathophysiology and better treatments that is not, it's using a human system. The problem most of the times we have been using animal systems to treat human brain, which are kind of very different from each other. So this is a system in between. Does that mean that it's better than animal models? Absolutely not, because I cannot use these organoids to actually tell motor function, to tell me what is affecting in terms of behavior. So both need to work together to help us to answer.
Yeah.
Going back to the circulating DNA, you asked me where they are coming from. So we're trying to use these models to actually look if the cells that are either dopaminergic or glutamatergic are releasing more of the circulating DNA. So which cells are more stressed in patients with bipolar disorder? This is an ongoing study in the lab, so I don't have yet data. So maybe when we have, I'll send it to you.
So you said the organoids will expand, do they divide? And you mentioned that they do have astrocytes. So it's not neuron specific. So the organoids grow out and they have like oligodendrocytes, astrocytes, and
So
other, okay.
yes, you're right.
Okay.
Independent, you can generate different types.
Mm-hmm.
You can generate more on 1 or 2 months where you only have very immature neurons and you have neuroprogenitor stem cells, but you don't get to a very complex organoid. We have right now in the lab currently about organoids have been kept growing in good conditions up to 10 months, and then you start seeing that. Yeah. You can go quite far, and you can actually start seeing that in astrocytes or oligodendrocytes, but it does take more time for them to mature and to work on this. In collaboration with two engineers, we are creating now a vascularized or system that helps to bring oxygen into the inside the cell. So we're probably going to be able to even mature further the systems and get better responses, but that is still some years to come to have an ideal system.
Yeah. I was thinking, I was thinking like what's the PO2 of the partial pressure
Yeah.
of oxygen in it. I did research on hippocampal brain slices and we use 95% oxygen, 5% CO2. And that's hyperoxic, but yeah, so you're limited, the organoid is limited in size and the core of that will be, they're going to have an O2 gradient. So that's very innovative that you're working with, people that to develop a vasculature to deliver oxygen to.
Yeah, so it's quite fascinating because it's a chip that has a vasculature, so microfluids, and this microfluids forms a kind of scaffolding, and the cells kind of grow around of that scaffolding of vasculature, if you will. right?
Yeah.
So that the now you can deliver it because of this, the nutrients, the oxygen in all the sphere of your organoid. So that will actually enable a much more precise oxygen tension across, and which we know very well that is crucial for interpretation of any of this type of data in brain
Yeah.
organoids. So a lot of the technology is promising. I think it will help us to understand better neurological, psychiatric diseases development. But it's still, every time I read it, I'm like, but you had a necrotic core in that
Yeah.
organoid,
Mhm.
right? So a lot of
Yeah.
things still need to be determined to interpret that properly. But it's promising. I think it is
Mhm.
really interesting that we're able to actually have a platform that can derive your brain cells with your genome that can help to understand what is actually better for your own brain cells,
Yeah.
not from
Mhm.
somebody else's.
Yep. I have some questions about mitochondrial DNA. And, so the mitochondrial DNA, from my understanding from cancer, is really vulnerable because it lacks the robust DNA repair mechanisms that you may find, for example, in the nucleus.
Right.
However, the fidelity of the nuclear genome is dependent upon the mitochondria making ATP because it's a very energetic process for DNA repair. But
Right.
the mitochondria has DNA, and that's like the site for reactive oxygen species production. So you have DNA juxtaposed to the electron transport chain, pumping out lots of superoxide and other reactive intermediates. So it's sort
Mhm.
of very,
Mhm.
and you have that, and you don't have the robust DNA repair mechanisms
Mhm.
of it. So in my mind, I kind of think of that. That makes the mitochondria selectively vulnerable to sort of these mutations and things that you're looking at, and that will be, you could be genetically susceptible to that. And I guess there's always a discussion that you get your mitochondria from your mom, and I'd like to discuss that a little bit more, but a little bit.
Mhm.
I'd like to discuss sort of and I don't know if you've looked at this, the vulnerability of the mitochondrial DNA relative to the nuclear DNA, and then the crosstalk between, we call it, I think, the retrograde response in cancer biology, at least. And is it true that we get our mitochondria from our maternal origin? So those
Yes.
two questions. Yeah.
Okay, let's start with the maternal origin because it's one of my favorite questions.
Yeah. Mhm.
So, yes, it is true. We inherit our mitochondria from our mother, and that's the mitochondrial DNA that comes from your mom, and that's passed the lineage from that. But you have to remember that this mitochondrial DNA only forms 13 proteins from your mitochondria. But the mitochondria itself has 1,400 proteins that needs to come from the nuclear DNA. So the
Oh, wait, really? I didn't. Okay.
yeah.
So if we talk about the electron transport chain, I
All,
know the complex two,
all
excluding
nuclear.
complex two, I thought all the other complexes were derived from the mitochondrial genome.
Nope.
Is that not okay? That's not right. I
Okay,
don't
so
know why I was
let's
thinking that.
so complex one, for example, yeah, it has 44 proteins. Out of these
Yep.
44 proteins, only I forgot exactly the number, but let's put like 4 or 5 of them come actually from the mitochondrial DNA. The rest comes from the nuclear DNA. The
Okay.
complex
Mhm.
two, for example, all comes from nuclear DNA. Complex
I
three,
knew complex two did. Yeah, I didn't
yeah.
know
So
complex one.
complex
Okay.
three, four, five, they
Yeah.
all have different subunits that have
Hmm.
genes that produce proteins that are transported into the mitochondria, the protein, to assemble the complexes itself. So
Okay.
this is so important is because when we initially thought that mitochondria only comes from the mom, so it's like, okay, it needs to be inherited from, right? Should be like, but that's called the rare mitochondrial disease, where the
Yeah.
mutations are incurred on the mitochondria DNA that is inherited from your mom. And those mitochondria, as you said, they don't have a mechanism of repairing the DNA. So how the mitochondrion maintains itself is by biogenesis, by combining fusion, fission, divided, and then actually trying to remove those that are bad and continue. If you accumulate a lot of bad mitochondrial DNA, it's called heteroplasmy, your heteroplasmic gain. If it passes a threshold, the rare mitochondrial disease are then become evident, right? But when we're talking about mitochondrial involvement in neurological and psychiatric disease, we're talking about the portion of the nuclear encoded genes coming into place to interact with this mitochondrial DNA and not assembling your mitochondria on the best efficient capacity. So mitochondria is functional. You don't have a rare disease. You're not dying of a rare disease, but you accumulate this dysfunctional link between your two that cannot form a mitochondria that works at the best capacity. It's like if you have a car that is supposed to run in one type of fuel and you put the wrong fuel. It might turn on for a little bit, but it's not going to run in the best capacity of that car.
Coffee overconsumption and caffeine dependency is no joke. So overconsumption of coffee and energy drinks that have caffeine are associated with an increased likelihood of anxiety and even alcohol consumption to take the edge off at the end of the day. People jacked up on these stimulants often look for things to calm down at the end of the day. So that's really unnecessary because we have many better options out there. If this sounds like you, if you're strung out on coffee or too much caffeine, it's time to try mud water. So this stuff will really give you a smooth energy without the jitters, without the irritability that often happens when you're overstimulated on coffee. So mud water is more than just a coffee alternative. It's loaded with adaptogens, antioxidants, and super high in polyphenols. Every single ingredient in mud water products are 100% USDA certified organic, non-GMO, gluten-free, vegan, kosher, and zero sugar. Add allulose. I tend to add allulose or a pinch of monk fruit to sweeten it. The product has cacao and chai, and that gives a little hint of caffeine and also has like more of a hot chocolate flavor. It's got lion's mane, it's got cordyceps, and this is really important for enhancing focus and attention throughout the day. It's got chaga and reishi too to support immune system health. So ready to make the switch to a cleaner and calmer energy, head to Mudwater, that's MUDWTR.com, and grab a starter kit today. Right now, our listeners do get an exclusive deal, up to 43% off the entire order, plus free shipping and free rechargeable frother when you use the code Metabolic Link. And the URL is mudwater.com/metabolic link. That's MUDWTR.com/metabolic link. That's right. It's 43% off when you use that code. After your purchase, they'll ask you how you found them. Please show your support and let them know that we sent you. Now back to our episode with Dr. Andreazza. And do we know, Anna, the biochemical sort of fundamentally, does it have to do with cardiolipin, like the immature cardiolipin to that? Is it, part of the inner mitochondrial membrane? Is it structural? Is this function tightly associated with a structural component? I think that's what I'm asking.
Yeah, great question. Yes, in some of the diseases, the answer is yes. In bipolar disorder specifically, as we've been talking a lot about bipolar disorder today, we don't have that answer. We do know from many postmortem brain studies that mitochondria electron transport chain subunits, for example, like NGF7 in complex 1, is downregulated in the transcriptome level and in the proteome level. So a lot of the associations we have seen from microarray studies and from transcriptome studies and the proteomics studies for patients with bipolar disorder, schizophrenia, and major depression are more in the assembling units of the electron transport chain, not so much in the cardiolipin or membrane pores that form the complexity of the stability of the mitochondria itself. Okay. There are some studies showing alteration in biogenesis of the
Hmm.
mitochondria, genes that controls the fusion and fission of the mitochondria. But that is less replicated in this field. More
Hmm.
into the Alzheimer's and Parkinson's, actually a lot on that area as well.
Yeah, I know in cancer, Dr. Thomas Seyfried has kind of done a deep dive into looking at like cardiolipin and immature cardiolipin and crystallysis,
Mhm.
the disassembly, if you will, or kind
Yeah.
of mitochondrial dysfunction at that capacity where you have almost a shell of a mitochondria and
Yeah.
it'll still take up oxygen, but it's not coupled to ATP production.
Yeah.
And then,
And
yep.
there is one paper comes to mind from Cataldo from Harvard. That quite a while ago, it was published actually in early 2010 or 12, something around that time, where he showed like a smaller mitochondria networks, and we replicated that later on in actually white blood cells of patients, showing again smaller mitochondria in
Hmm.
patients, and now we have this data replicating in organoids.
Hmm.
So it seems to be the case that there is actually something that is actually making the mitochondria to either has a less efficient capacity of elongating, which is extremely important, especially in the
Yeah.
brain, to become efficient on oxygen coupling and the energy production. So it seems to be that's where it's going on the
Yeah.
field, but we don't
Hmm.
have
Yeah.
I don't know, can't
Hmm.
say the word
Yeah.
probably.
I know what you mean. Yeah.
Yeah. Uh,
Crystallography, maybe? Yeah.
Crystallography. Yeah,
Yeah.
thank
Yeah.
you.
Yeah.
We don't have that actually
Okay.
have data, and it would
Yeah.
be beautiful if someone can do it
Uh-huh.
because
Yeah.
a lot expensive, and but it would be
Hmm.
beautiful to actually getting that depth of details.
Something just popped into my mind, and I've always wanted to ask a mitochondrial expert about this because I did studies more than 15 years ago on
Mhm.
isolated mitochondria from the liver. It's super easy to get it from the liver, and then the brain. And then I was looking at these isolated mitochondria in a hyperbaric chamber using atomic force microscopy and laser scanning confocal microscopy. And with the AFM experiments, what I observed was these mitochondria like self-assemble into these little rings. And I was pulling my data off of the old computers like 2007 or 8. And I was like, I always meant that it's like the mitochondria would self-assemble and there'd be a whole bunch, and there's all you would see
Yeah.
when I would scan it with an AFM was these rings of
Yeah.
mitochondria, and they would just self-assemble like,
Yeah. It's,
and
it's, it's, it's quite phenomenal. Liver mitochondria especially has this capacity of like merging and can actually connect into each other because
mhm.
you produce on the liver the Cori cycle, and you need that very efficient mitochondria to actually get out and in and out of the state of glycolysis, and moving into a state from another. So they are
Mhm.
really, really efficient. It's like if you're looking a little bit like amorphosis of a
Mhm.
cell to another. But it's very different. And this is such a beautiful point you make, Dominic, because our mitochondria in our body, it is really tissue specific on actually trying to provide the best capacity you need for the muscle mitochondria, for example, is a much more stable mitochondria, is an elongated mitochondria, because you need to actually have more glycolysis, because you need to get that easy glycogen and easy pulling out and moving fast energy for your muscles to contract very, very quickly. But a
Mhm.
mitochondria in the brain is a very different mitochondria. It needs to be
I
fully,
noticed that. Yeah,
right?
it's very with all the lipids and everything, it's like very hard to get isolated. Where the liver is so easy, but
Yeah, you
so
get like a lot of them, right? And
yeah,
it's
yeah. Hmm.
beautiful. But if you take it, this is a quite an interesting new area, and it's called mitochondrial transplantation.
I had a
So
question about this.
yeah,
Yes.
so
Yeah.
maybe we enter on
Mhm.
that now.
Yeah, yeah.
So
That
this
was
is
my next
actually
question, actually. Yeah.
a really cool field that is studying, and we are
Mhm.
diving into it because you can regenerate organs by actually transplant mitochondria. So one of our collaborators have done this study where he took mitochondria out of liver and he wants to regenerate lungs. And initially we thought that, it's not going to work because the mitochondria from the liver is so different from the mitochondria of the lungs that they're not going to adapt. And actually not that the case, because the mitochondria has such a beautiful adaptability to the oxygen tension and environment of itself, that in adapting the shape of the mitochondria of the lungs become a highly efficient mitochondria to do coupling of oxygen and regenerate the lungs on itself. So that's not my lab experiment, somebody else's experiment.
Yeah.
But it's such an interesting area that you now starting to understand more. You can actually take mitochondria, extract the mitochondria, purify, and transplanting cells to help your cells to regain the function of a better capacity, energetic capacity. Where this
Quick question. If you were to take your
Yeah.
own mitochondria, if you were to take somebody else's mitochondria,
Great
but also a quick question about the liver, because my understanding is that liver mitochondria lacks succinyl-CoA transferase. So the liver can make ketones, but it can't use ketones as an energy source because it lacks, I believe, that enzyme. So ketones get into circulation. The liver is very greedy. Like whatever you eat, it kind of sucks up and uses, but it is the producer of ketones, but it doesn't unless you have a liver cancer. I guess that would be transformed. So, but what you're saying is that the liver is the mitochondria are highly plastic and adaptable to whatever environment.
Yeah, we can adapt,
transform.
but what we don't know at all, and I don't think this group that is doing this study currently knows at all of this, the answer to that. You know that the mitochondria enter in the system and is able to recuperate the system, give the energy that it needs. But is that mitochondria staying? We don't know. Is that mitochondria being autophagied out of the system? Do we gonna need it to redo it again, the transplant, to maintain? So you asking again the question, can I use the mitochondria from different tissues from myself to myself, which is called autologous, or myself to somebody else, which is called heterologous, or allogenic. People use sometimes interchangeable these words. Or if you taking from an animal to a human, that's called xenogenic, right? So all these questions are open questions in this field of new mitochondrial transplantation. And I think it is an area that in the next while will explode in understanding better, could become a therapy. I would think that the first targets of therapies for this would be regenerative medicine, and then we'll probably see this evolving into probably acute states, and then chronic diseases in the later stage, because a lot to be learned. Really, really new, but it's fascinating. If you ever want to see one of my favorite videos, go to James McCulley website. He's the creator of this technology in Boston. And he has a video that shows the mitochondria being injected and engulfed by a cell. It's fascinating to see that that is plausible, and we can one day be using that to regenerate retina muscles.
The retina, yeah, for sure. Yeah. So, okay, so there's mitochondria replacement therapy, and then also mitochondrial restoration therapy, I guess, and mitochondrial therapy. So maybe talk about the state we're in right now in regards to mitochondrial therapies, and we could talk maybe about certain antioxidants, urolithin A. We just did a podcast on that, and that was interesting. So, would love to hear your perspective on the current state we're in right now with mitochondrial restoration.
So that is mitochondrial transplantation, which we just talked. Very, very new, lot to be learned, needs to be better understand, to know where should be used, when should be used, and how to be used. Mitochondrial replacement therapy that has been studied in the UK, there is Mexican studies we don't have yet in Canada. I'm not sure yet if you have in the United States, but that has been successful in delivering novel babies. So basically what this is replacing the mitochondria of a novel with the mitochondria of a donor, and fertilizing with the sperm of the dad, and then reimplanting on the mother. So the mother was a carrier of a defected mitochondria, and now is being replaced. And it's been very successful. It's over 10 years now. Babies are actually very well and successful and growing beautifully, but a lot of ethical debate around that. So not sure when going to be coming, unfortunately, to Canada, to US. There is lots of the ethical debates to be solved before we can actually call this as a curative therapy for mitochondrial disease. But it certainly brings hope for a lot of patients and a lot of women out there that carries mitochondrial disease, a rare disease. Mitochondrial augmentation therapies or mitochondrial restorative therapies. So we can talk about the augmentation as something that improves the mitochondrial biogenesis. So you have many of the compounds out there, like bezafibrate, that improves PGC1 alpha and gets the mitochondria actually replicating more. Then you have the antioxidant system that you were talking. And you know a common friend of ours, Mark Tranopolsky, and Dr. Tranopolsky has numerous of antioxidant combinations that are highly effective for improving mitochondrial functionality. So again, for bipolar disorder, difficult question on antioxidant therapies, because we have to be a little bit careful to not enhance the mitochondrial ATP production to a point where you're flipping the system from depression to mania. And there is some cases where we know that NAC has been unfortunately pushed from depression to mania. So I think we need to understand a little bit better when to use, when to wash out, and when to target for therapies for better. So back to the beginning of the talk, find the pathway that is involved on you will tell you what is the treatment that you needed for the patients.
Yeah. And what you're really advocating for in mitochondrial or metabolic medicine is the use of multi-omics, metabolomics should be like part of, we get our blood work and blood tests and things like that, but we can gain more insight into, the simple one is, glucose and then lactate, as you mentioned, but that you envision a future where mitochondrial medicine will incorporate metabolomics and other multi-omics approaches.
I hope so. I think that will be my final message for anyone that is listening, is like, if we needed to put our minds together to solve that puzzle that it can come together, is really to find a way to the future to provide sustainable testing, clinical testing, because the problem with a lot of the omics that we produce is so expensive that is inaccessible, and then we end up never putting them in clinical translation. So can we find our minds to work together to find processes where we can really define biomarkers that can become clinically relevant and useful and sustainable to become incorporated, because it has to be easy for the doctors to also be able to understand and to take the blood of the patients and not be owners for any part. Because if we put more ownership on anybody out there, we end up delaying translation. So I would love to see our minds working together to get to that point where I believe is possible.
Yeah, an evidence-based metabolic, platform where, an assays that can be developed, a lactate, you've proven, there's so much evidence just in that alone, but we know there's so many other metabolites that are key.
Exactly.
And with keto and how these change in response to ketogenic diets that enhance mitochondrial function, lower lactate. So yeah, a lot of work to do, and I know you're
Yes.
spearheading it, and we thank you for all the work you're doing. I know you are spearheading the Mitochondrial Innovative Initiative, Mito 2I, I think you call it, is the abbreviation for
Yes.
that. I would love to hear what you're doing on that front.
Yeah. So the website is www.mito2i.ca, and this basically is a free platform for bringing community together. The story behind is as I studying bipolar disorder, I stumble on families that actually have patients within the same families with mitochondrial rare disease and patients with bipolar disorder. And the mother approached me and asked me such a simple question: how these two diseases are connected. And I had
Mhm.
no answer to give to her. That was like, wow, I'm sure there is a genetic profile here, but I don't know how. So we start working together with these families, and that become open a huge curiosity for us why we don't have initiative or a network that actually bring people from cancer, from bipolar disorder, from rare diseases, from Parkinson's, from any type of health and disease that is studying metabolism and mitochondria, and just let's put them all together and try to see if we can understand mitochondrial metabolism in health and disease. And through that, perhaps we can apply for not only one disease, but for many diseases and learn from
Mhm.
each other. Like cancer, we know so much on mitochondrial medicine in cancer, and we know so little in other diseases. So how can we learn from
Mhm.
each other? So that's
Yeah.
basically what it is a platform to bring people together. We fund a lot of graduate students. We have um, opportunity for anyone that is around the world to come and join a training session where we bring an expert, and that they will actually go and dive in for 3 hours on how do you measure the ATP in the cells, in the brain, in the liver, which technique you should use to measure this. And then in the next training session, it might be a clinician that is going to say, here is how I diagnose mitochondrial disease, and walk
Mhm.
through. And then we have a debate with another neurologist to see what is going. So check it out. We do a lot of collaborative programs, and we love to be working more with you at Metabolic Link and see where we can take this further.
Thank you for doing that. Yeah, the implications are tremendous. I mean, cardiovascular disease, neurodegenerative disease, cancer, they all are essentially metabolic diseases.
Right.
And, it just goes across all the different health maladies that we're dealing with. So thank you so much for
Thank you.
spearheading this work, for the work that you do, for sharing your knowledge, your wisdom and expertise in mitochondrial medicine. And I look forward to following your work and hopefully collaborating and hopefully
Yes.
seeing you soon.
Yeah, I hope
So
so
much.
too. And I hope you will come to Toronto to see us in here.
I would be honored. I would love to. Thank you.
And thank you very much for having me here today.
Thank you, Anna. Thank you for watching and listening to this episode with Dr. Anna Andreazza on the critical role of mitochondria in mental and metabolic health. If you like this episode, please be sure to share it with others who might benefit from learning about the potential of mitochondrial medicine. Don't forget to subscribe to the Metabolic Link on YouTube for more deep dives into metabolic health and the groundbreaking research that we like to share with you. And please leave us a review on whichever major podcast player you use to tune in. And if you'd like to access to exclusive ad-free episodes on the Metabolic Link, along with hundreds of other interviews, presentations, and content on metabolic health and therapy, please check out our medical education platform. That's the Metabolic Initiative at membership.metabolicinitiative.com. Thank you again for listening, and we'll see you next time.
Do you love learning about metabolic health? So do we. It's why we created the Metabolic Initiative, an online educational platform providing evidence-based education on metabolic health and therapies for healthcare professionals and the general public. By joining the Metabolic Initiative, you'll gain access to hundreds of expert lectures, interviews, panel discussions, and even private episodes of the Metabolic Link. CMEs are available. Go to metabolicinitiative.com to get started. And as always, thank you for listening to the Metabolic Link.